Hey, party people, and welcome to the Scriptome podcast, where we discuss the AI-based tools that biotech researchers use to get the job done and get therapies to market. I'm your host, Stu Angus, and today we're diving into Profluent.Bio's groundbreaking work on AI-designed gene editors. Last week on the podcast, we talked about bolts, how AI can now match nature by predicting protein structures. This week, we're talking about something even more progressive. AI that creates entirely new biology from scratch. What if instead of hunting through bacteria for millions of years of evolution, we could just design better gene editors on demand? That's the bet Profluent is making with OpenCRISPR-1, the world's first AI-designed gene editor. With me today is my colleague Pavlov, who's going to help me unpack this technology and what it means for small biotechs. Let's do this.
Party on, Stu. So let's start with the hook. Last week was about an open source AI tool called Bolts that has the ability to reproduce nature. This week you're saying AI is designing new biology. What's the difference?
Great question. Boltz showed that I can predict what a protein looks like. Basically, AI is reading nature's blueprints. But Profluent is doing something different. They're writing new blueprints. They're generating gene editors that evolution never created. It's the difference between copying a design and inventing one.
Before we get into the AI part, can you remind listeners what CRISPR is and why it matters?
Absolutely. CRISPR-Cas9 won the Nobel Prize in 2020 for a reason. It revolutionized biology. It's basically molecular scissors that let you cut DNA at precise locations and edit genes. The tool has changed medicine, agriculture, and basic research. But, and this is important, Cas9 isn't perfect. Cas9 is a big protein, which makes it hard to deliver into cells. It has off-target effects, meaning it sometimes cuts the wrong place, and its performance varies depending on the cell type you're working with.
So how have scientists traditionally tried to improve on Cas9?
The old way is basically a treasure hunt. You screen thousands of bacteria, looking for natural variants of CRISPR proteins. You sequence the candidates, test them in the lab, and hope you find something better. It takes years, and you're limited by what evolution already created. You can't ask for specific properties, you just get what nature gives you.
I guess this is where Profluent.Bio has stepped up. Who are they, and what's their approach?
Profluent was founded by researchers from Salesforce Research and DeepMind, so they came at this from an AI-first perspective. Their core question was, can AI design better gene editors instead of waiting to find them in nature? They've raised $75 million from top-tier VCs because they're not just making one-off editors. They're building a platform for on-demand gene editor design. That's an infrastructure play, too, like we mentioned with Boltz last week.
Walk me through how their AI actually works. How do you go from training data to a functional gene editor?
4 steps. First, they train AI models on millions of CRISPR protein sequences from nature. The AI learns the patterns of what makes a functional editor. Second, the AI generates novel sequences. These are proteins that don't exist in nature, but should work based on what it learned. Third, and this is critical, they validate everything in the wet lab. You can't just trust the AI. You have to prove it works in real cells. Fourth, they iterate, they test performance, feed the data back into the model, and improve the next generation.
So what did they create? What is OpenCRISPR-1?
OpenCRISPR-1 is the first AI-designed gene editor to hit the market. It launched in June of 2024, and here's what matters. It has comparable editing efficiency to Cas9 in validated cell lines. So it's not just a proof of concept, it actually works. But the big deal isn't just performance. It has 3 major advantages. 1, it's a novel sequence, meaning it's in a different intellectual property or IP landscape than Cas9. More on that in a minute. 2, it's fully open source. Under an Apache 2.0 license, anyone can download it, use it, modify it, no permission needed. 3, it's available today. You can go to Profluent.Bio right now and grab the sequences, plasmid maps, and technical docs.
You mentioned IP. Let's talk about the patent situation with CRISPR. Why does that matter?
CRISPR patents are a nightmare. You've got the Broad Institute versus UC Berkeley in this long legal battle over who invented what. Classic East Coast, West Coast rivalry here. If you're a small biotech trying to use Cas9, you're looking at expensive licensing fees and complicated negotiations. OpenCRISPR-1 sidesteps all of that. Because it's a different sequence, it's different IP, and because Profluent released it under Apache 2.0, there are no licensing fees. For a small biotech that doesn't have a legal team or $1 million budget, that's huge.
So this is truly creating, not just copying. How is that different from, say, mutating Cas9?
This is not taking Cas9 and changing a few amino acids. It's not finding natural variants. It's generating entirely new sequences from scratch. The AI is designing biology that nature never made, optimized for specific properties. That's the paradigm shift. You're not limited by evolution. You can say, I need an editor that works better in T cells, and the AI can try to design one.
Let's get practical. If I'm a small biotech, how do I actually use OpenCRISPR-1?
It's the same workflow as standard CRISPR. You design a guide, RNA, that's what tells the editor where to cut. You use the OpenCRISPR-1 plasmid instead of a Cas9 plasmid. You deliver it to your cells using standard methods, electroporation, lentivirus, lipid nanoparticles, whatever you'd normally use. Then you assay editing efficiency. You're basically swapping out the editor and seeing if it performs better for your application. Profluent has the full sequences, plasmid maps, and published validation data on their website. You can order the plasmid from a DNA synthesis company and start testing it in your lab next week.
Can you give me a real-world example of where OpenCRISPR-1 might be better than Cas9?
Sure. Let's say you're doing CAR T cell engineering. You're editing primary human T cells to make them attack cancer. The challenge is that T cells are hard to deliver into, and off-target edits could be dangerous. With OpenCRISPR-1, you could test whether it has better specificity in T cells. Maybe it's smaller, so delivery is easier. Maybe it has fewer off-targets, and because there are no licensing fees, you can afford to run those experiments without a huge upfront cost. The open source license also gives you flexibility. If you need to optimize the editor for your specific use case, you can modify it. You're not locked into a vendor's product.
What are the broader implications here? Why does this matter beyond just having another gene editing tool?
3 big reasons. First, cost. No licensing fees means small biotechs can compete with big pharma on tools. You're not priced out before you even start. Second, speed. Instead of waiting years for nature to give you a better editor, you can generate candidates on demand. Profluent is building a platform where you could theoretically request custom editors for specific applications. Third, customization. You can design for specific properties, size, specificity, cell type compatibility. It's like going from off the rack to custom.
Let's talk applications. What kinds of things can people do with this?
Pretty much any gene editing application. Gene therapy, clinical precision editing for genetic diseases, cell line engineering, if you're making biomanufacturing cell lines and you need stable, predictable edits, agricultural biotech, crop improvement, research tools, any academic or industrial lab doing gene editing. The key is that it's general purpose. You're not locked into one therapeutic area.
Where does Profluent go from here? Is this a one and done, or is there a roadmap?
They're just getting started. OpenCRISPR-1 is the proof of concept. They're building a platform to design base editors, prime editors, and transcriptional regulators, basically the whole toolkit of genome engineering. They've raised $75 million, and they've got backing from top VCs who understand the long-term play. They aren't really a direct competitor to Boltz, but they are similarly going for infrastructure for the next generation of biotech.
If someone listening wants to try it themselves, what should they do?
4 steps. 1. Go to Profluent.Bio and download the sequences and documentation. 2. Read the technical papers so you understand what's validated and what's not. 3. Order the plasmid and benchmark it against your current system in your cell type, your application. 4. See if it works better for you. Don't take anyone's word for it. Test it. That's the beauty of open source. You can evaluate it with zero risk.
Bringing it back to your consulting work at Scriptome.AI, how do you help small biotechs evaluate tools like this?
We help teams separate signal from hype. A lot of companies get pitched AI tools from vendors that sound amazing but don't actually solve their problem. We help you figure out, is this tool validated for your application? What are the real-world limitations? How does it integrate into your existing workflow? Is it worth the time and money to adopt? It's quite comforting for biotech clients to know that we are impartial. and unbiased when it comes to recommending tools without having to deal with the sales pressure or short trial period to figure out if it's worth it. For tools like OpenCRISPR-1, we'd help you design the benchmark experiment, interpret the data, and decide whether it's worth switching over. We're basically your in-house AI evaluation team, but without the full-time hire.
Final thoughts. What's the big takeaway for listeners?
AI is moving from prediction to creation. We're not just reading nature's blueprints anymore, we're writing new ones. And because Profluent made this open source, small biotechs have access to the same quality tools as big pharma. If you're doing gene editing, you owe it to yourself to at least test OpenCRISPR-1. If you're thinking about AI in biotech more broadly, pay attention to this pattern. Open source is leveling the playing field. That's it for today's episode. Feel free to reach out to us or the team at Profluent.Bio to learn more. And let us know what you think of OpenCRISPR-1 in the comments below if you're able to try it. Remember to hit subscribe if you're interested in keeping the party going and hearing more biotech tool insights and reviews in the future. I'm Stu Angus, and this is the Scriptome podcast.